So, as a first speaker, I would like to welcome Dr. Halenur Yavuz-Kienle from SYNLAB, MVZ, Humangenetik, Mannheim. Halenur completed her PhD in 2014 in Göttingen and has a some years of experience as a clinical scientist. Since 2018, she has joined the SYNLAB MVZ in Mannheim, and today she presents the strategy of analyzing WES cases and how varvis helps with this. Thank you for agreeing to speak here, and welcome, Halenur.
Thank you, Yvonne. It's my pleasure. And I'm very excited to be present here today. After 2 years, I'm looking forward to these exciting 4 days, and I hope you do too. And I want to kickstart this session by introducing to you some of the cases that we were solving using the varvis software, what kind of strategies we used, and demonstrate this with the case examples that I brought with me today. We are a diagnostic lab, part of SYNLAB, situated in Mannheim. We have also, two parts of the laboratory. One part is the medical practice. We have genetic counseling sessions, so internal cases, internal probes come from those sessions. But we also receive external samples. The second part of the laboratory is where we perform the analysis, write the medical reports, and discuss many cases like I'm going to show you today. We are an accredited laboratory performing whole exome sequencing, mainly NGS analyses. Big panels we also analyze with the focus on cancer genetics, cardiolipid genetics, but also syndromal diseases. I am leading a team of 5 scientists in the center who are mainly responsible for whole exome sequencing analysis. And the strategy we follow using varvis software is, I can summarize it mainly with these 4 points. Varvis provides you with the mode of inheritance predefined filters, so you can go ahead and start your analysis with these predefined mode of inheritance filters. If you have a given pedigree, relatively clear family history, let's say you want to check for X-linked or autosomal recessive disorders, that you can do right away and start your analysis with that. But we also look for variant annotations, the coding effects, for instance, like frame shift variants, we would like to filter before we analyze all the missense variants. Population frequency, like filter out very frequent variants. We also use this nice feature now newly available on varvis that you can also enter for a given specific variant or a gene. You can also enter the literature specifics to this variant and then filter against that if you want to. Of course, relevant databases information like ClinVar annotations, for instance, are also available in the software. So we do this also, like specifically search for this also. But if we didn't identify any certain variant using these 3 strategies, then we really look at the patient's phenotype, of course, and try to have an overlap with the called variants, gene phenotype associations, and see which variants that were called for the patient's sample is overlapping the best with the given phenotype of the patient. Okay, so the first example I brought to you is of a 7-year-old patient with suspected Noonan syndrome. Like I said, here I entered the de novo filter. On the right-hand side you can see, and then I basically filtered for good quality variants that are rare in the patient. And I think I sorted them against the HPO similarity score, and one of the top variants is an LZTR1 variant, a missense. If you know this disorder or syndrome well, you know that gain of function variants are described for Noonan syndrome. And so once we saw also that the variant was only present in the index, so mother and father didn't have the variant, we were reporting for the patient a likely pathogenic de novo variant, confirming the diagnosis of LZTR1 related Noonan syndrome. Second case where we used another mode of inheritance filter was this patient with developmental delay, splenomegaly, anemia, dysmorphism, and this was a family that had consanguinity. When we filtered for autosomal recessive variants, because the family is consanguineous, right? So we wanted to see how many or what kind of homozygous variants we identify in the patient. We come across two top candidates in PEPD gene and CTC1, and we actually were not, they have comparable HPO similarity scores, so we chose to report both of them and as uncertain variants. And in our report, we actually mentioned that the PEPD deficiency can be also, if the diagnosis is suspected, can be further analyzed, can be further researched. Later on, from the case after reporting, we received feedback that this was done. Actually, after receiving our report, they did the biochemical analysis and actually confirmed a prolipidase deficiency. Upon this extra information, we could use another ACMG criterion that was not used initially, so we could classify the PEPD variant. I think now in the system it's a likely pathogenic variant. Last case where I would like to show you the mode of inheritance aspects of filtering is a case where I brought two CNV to show you. So here I'm not using any filter for mode of inheritance, but you can actually look for large deletions, duplications in the index, but then also list these data of the index and mother and father under each other, so you can see nicely that the index actually had two deletions in the auto-A gene. The patient had the suspected diagnosis of bilateral hearing loss. So compound heterozygous deletions in the auto-A gene actually confirmed the diagnosis of the autosomal recessive type 22 deafness in the patient. So the father here in the bottom bottom line, you can see the father has a large deletion in this gene, but the mother has an exome, single exome deletion. So the child, unfortunately, has a homozygous deleted area, and that is actually diagnostic for the child. So, mode of inheritance we talked about. Here is a variant, for instance, in a patient, in a fetal sample with abnormal nuchal translucency, and later on with the next ultrasound findings, furthermore fetal malformations presenting with. We see this frameshift variant and nothing else in the patient, but it's paternal. POC-Z gene is known for de novo, mainly de novo variants described in the literature. So having a healthy father carrying this frameshift variant that is described in the literature also doesn't fit the mode of inheritance pattern that we are expecting from this analysis. Nevertheless, because there is a good phenotype match, we discussed this in the report and reported this variant, hinting that there needs to be a clinical re-evaluation in the family done, which was the case later on. Luckily, it was an internal case, so we could get the feedback afterwards that the childhood pictures of the father even showed some mild symptoms of the White Sutton syndrome. He was mildly affected. I think I didn't do the follow-up on this case, but I think later on we received family members. I'm not sure if we confirmed it in the father as de novo, but it, of course, can be done afterwards. Right. There you can see that the variant annotation, as the variant was described in the ClinVar once as uncertain significance and once as pathogenic. You can also see the phenotype, how much to a certain extent of the patient and the POC-Z gene is matching to the given HPO terms. Remember the very first case, the de novo LZTR Noonan variant? Let's say we missed this variant during our de novo search or during our mode of inheritance search. We missed this variant, we didn't see it. we would then catch it during our third filtering strategy, because the variant is also known to clean our database, twice uncertain, 3 times likely pathogenic, twice pathogenic. So in case one misses the variant due to mode of inheritance filtering or variant annotation, later on you can also catch the variants. It's then the advantage of using different strategies, different filtering strategies with this software. So if one missed the de novo variant, then they would have to see the variant while they are searching for the variants that were described in the literature afterwards. Like I initially mentioned, you can even then enter which PMID is exactly describing this variant for the Noonan syndrome. If this variant, by the way, were not to be written, described in the literature, you could still, maybe the neighboring amino acids have been described before, you can still leave your comment on the given PMID for the neighboring variants for the gene. So it doesn't have to be for the variant, but for the gene, you can also document it in your case. This part is what I like the best, because if I didn't identify any straightforward variants, right, a de novo variant is easy to find, a homozygous variant in a consanguineous family is easy to identify. If we didn't see anything so far in a case, then becomes the more interesting part for us to see. This is a 31-year-old patient with an initial suspected diagnosis of Gardner syndrome. We received too little information on this patient initially. Only these, I think, yeah, this was all it. I think we received 4 HPO terms on the patient. And once we did our HPO similarity score match, so that's what basically what we are doing with the phenotype-based search. So we are looking at the HPO of the patient and then HPO related with the LEFR associated syndrome. Once we varvis does it for you, it compares these two in a matrix, and then the result is this HPO similarity score. Once we look at the rare variants in this patient that had the top HPO similarity score, the two hits were not really disease-relevant variants, but this LEFR -4 homozygous splicing variant was actually an interesting one. It was not really described in the literature, but it was causing in silico, according to the in silico parameters, it was causing a splicing effect. It was not, like I said, a known variant, but we reported this. It was our best candidate for this exome case. It was a solo, I think, and we reported it with, again, hinting towards clinical reevaluation. It was done later on, and I think in the meantime, as we reported this variant, they received new clinical reports on the patient. I think the doctor responsible for this case was then so convinced by this phenotype. He brought so many more clinical reports to us and asked for a reclassification report because he thinks now the phenotype match is so specific. If you look at LEFR clinical synopsis, the patient now fulfills many more overlapping features of this disorder. So we are going to classify this variant also higher. Another case where we were searching for a variant and not finding it, maybe internal tip for varvis users, already varvis users, HPO similarity score here brought this variant, ACTG2, for us, but not the HPO match, so be careful. This is a fetus with cloacal malformations and lower urinary tract obstruction. We did our search. I think the clinical scientists didn't even identify a variant in this case. We were about to report it almost negative, then we were discussing so specific phenotype with ultrasound findings. And then the clinician actually saw this HPO similarities, the ACTG variant with a high HPO score, abnormality of the lower urinary tract, abnormality of the anal atresia. This fits actually so much to this phenotype that we eventually ended up reporting the variant. This is a bonus case I brought with me. What happens when we don't find anything? Using these 4 strategies, what do we do? What else can we do with varvis? This was a recent case. A colleague of mine was analyzing a year-old patient's data with premature birth, severe hypotonia, and there was a suspicion of neuromuscular disease. She did a trio exome analysis but didn't identify any variant. We unfortunately received too little information on the patient, so she didn't know if there were any pre-analyses done on this child with severe hypotonia. She had the suspicion, she was just wondering, oh, I wonder if there was Prader-Willi syndrome excluded in this case. And she searched for rare variants in the patient on the 15th chromosome and just noticed that father doesn't have any variant called on chromosome 15. I mean, not the variants that are shared with the index. Of course, the father has variants called on chromosome 15, but they are not shared with they are not inherited to the child. So when you actually search for rare variants on the index, you see that they have been exclusively inherited from the mother. This was an enough indication for her to actually activate a methylated MLPA analysis. It revealed hypermethylation and a diagnosis of a Prader-Willi syndrome. Funnily, I was also busy with another prenatal case back then, and methylation analysis, MLPA for Prader-Willi, was actually activated for my case in parallel. Until I was analyzing the case, when I was finished, I didn't identify any variants in my trio exome prenatal analysis, but the methylation analysis was already finished and showed hypermethylation. So the other way around, like this case, I went back to the 15th chromosome and saw a very similar pattern to this case. So you just need to be curious. Using varvis, actually, even though you don't follow the strategies that are there for you, you can also even identify such interesting patterns using the exome sequencing data. Yeah, this is the team behind these cases. Already old, this photo, I have to say, growing. A big team. We are very dynamic and yeah, hoping for more questions and interest from you. Thank you for your interest.